Okay. Dzień dobry. Good morning, everybody.
Good morning.
We have
Good
about
morning.
40 people right now, so we can start. And I'm trying to remember we've been in which moment we stopped last time, but we were talking about. I think we were talking about cytopathic effect. How are you? Are you okay after the break? Did you have time to go to your family for a while?
We're fine.
Okay. Well, this is a hard time. I mean, everybody is worrying about the growing number of infections and the growing number of people in hospitals. I hope it doesn't concern our community, because both people in the institute and the students are vaccinated. I hope so that practically everybody, but still it is around us, and well, I'm constantly asking about that, and I'm also constantly asked about my opinion, why people are so against the vaccines, and sometimes I wonder, what is your opinion about it, because probably we have completely different point of view. So sometimes I would like to ask you that question, what you think about why people in your generation belong to so-called anti-vaxxers. Okay, but let's go back to viruses. I will try to open the last slides from the last presentation. Share the screen. That should be this one. So, can you see my screen?
Yes, we see it.
Okay, perhaps I already shown that one, but I just want to come back to this topic we finished when we were talking last time. Okay, that should be bigger. So I was talking about the effect which virus causes in the cell culture, and this example is of hamster embryo cells, one of very common cells used in virology. And you see here the uninfected cells and cells which are infected with the polyoma virus. This is virus which has oncogenic potential. That means that apart from killing cells or changing their shape, that virus is transforming cells into cells which have the same characterization as tumor cells. And here what is characteristic, that they can now grow on top of each other. That means that they don't have that contact inhibition which normal cells have. Another example of cytopathic effect, this is more familiar for me because this is something we see every day in the laboratory working with herpes viruses. This is the beginning of the plaque. I probably shown it to you, but just to remind you what we are talking about. It. So you can see the cells which are growing close to each other have that kind of shape, a little bit like bricks. The rounding, the touch from the surface of the vessel where the cell culture was growing, where the cells were growing, and then finally there will be a big hole in that culture. So I already showed also the effect of the amount of the virus, the number of the virus particles, the uninfected culture. Then this culture infected with small amount of viruses, which you can see single plaque, and with a lot of viruses, when the situation which each cell infected, the cytopathic effect is very, very clear. I will come back to that in a moment. So another examples of uninfected and infected cells. You can see that very often we can see just from the morphology of the cells that they were infected, and some of the effects of those cytopathic effects are very characteristic. Perhaps I will go. So I already talked about transformation. This is another example of cells growing on top of each other, but and another examples of plaque. But I want also to show you that sometimes there are some other effects. For example, here I already mentioned those rounded and detaching cells, but there is also all kind of inclusion bodies which can be observed in infected cells. Sometimes cells seem to fuse to each other because proteins which are present in viral membranes have that effect. They like gluing cells together, and we call it that formation syncytia. This is very characteristic from some viruses. For example, talking about inclusion bodies, this is so-called negro bodies. That means that this is actually used for diagnosis of rabies, virus of Chiclizny. So we can see that there are inclusion bodies, and there are some wytrącanie się czegoś. This is maybe stained with characteristic dyes, and this also is a way to recognize the virus. This is another inclusion body by herpes virus. That is what we are working with. And this is a few words about the intracytoplasmic inclusion bodies. Well, this is, the important is just to know that there are different type of inclusion bodies, and they may be characterized by staining with different dyes. So the inclusions can be small or big. That's typical sensitivity against that is HIV, and cytomegalovirus virus is really the name of cytomegalus is from the enormous large cells which are characteristic for CMV. I'm showing this all of that to you because I wanted to show you that for experienced virologists, experienced clinician also, seeing the dyed tissue from infected organism may be sometimes enough to recognize characteristic virus. Okay, so coming back to the inclusion bodies, this is in Polish, but this is just you can see in the image what is shown here. That if this is a cell and a nucleus, so the inclusion bodies can be present in the cytoplasm or inside the nucleus, or both in nucleus and cytoplasm. Sometimes the other. So this is one type of cytopathic effect. The other is that syncytium, that zespolnia, that fused the cells fused together, transformation, and lytic effect when cells are dying. So this has all kinds of cytopathic effects. And coming back to the inclusion bodies, what are inclusion bodies? Are usually there are the viral proteins which somehow accumulates in certain areas, or maybe also wrongly folded cellular proteins which are also somehow like precipitated inside the cell. So this is all this morphological effect are helpful in the diagnosis. Here you can see two different plaques by the same virus. This is herpes simplex, virus of Ryszczki. Here, and you can see that one is look completely different from the other. This depends on the feature of the characterization of the virus and also all cells. So, as I say again, morphology of plaques is important. And that brings me, that's about all what I wanted to say in the last lecture, and I will close this one. Just a moment. And okay, and then we'll open the next one. Okay. Mm-hmm. Okay. So now coming to more to diagnosis of viral diseases. I will have to wait for a second to open that. Yes, you can see that slide with diagnosis of viral diseases.
Yes.
So let's go on with that. So this is the slide just showing about diagnosis of viral disease. I'm now thinking about the clinic, not in the laboratory, but what can be done in the clinic. And the clinic in the doctor office, of course, clinical science are the first and most important features which are important to recognize viral diseases. But unfortunately, many viruses are are very difficult to recognize just by clinical signs, but the common cold can be caused by many viruses, from coronavirus, rhinovirus, adenovirus, and so on. But sometimes it is very important which virus disease it is, because for some we still have possibility to cure, with others we just have to take care of the patients. So this clinical signs, but of course there are very specific methods of virus detection. I will be talking now more about clinic than about laboratory, but this is very close. So we can of course isolate the virus and grow it in the cell culture, and using specific antibodies we can recognize the virus. We can also estimate the number of the virus by plaque assay and other methods which I explain in a moment. We can detect nucleic acids of the virus, for example by PCR, by polymerase reaction. We can use typically virology methods like hemagglutination, and I will explain that also. ELISA test is used both in clinic and in laboratory, and all kinds of immunological, immunochemical methods are used every day. Electron microscopy, I will be talking about it in a minute. So okay, I was trying to make you more familiar with some words, some terms which we use every day in virology, and that cytopathic effect I already explained a few times, and and changes which are introduced by the virus to cell culture. But you have to remember that not every virus is able to give that visible effect. Many viruses can grow in the cells and completely and stay completely invisible, at least in the microscope. But there are also ways to detect such a virus. The important for virology is virus quantification. That means counting the number of the viruses in specific volume. Now, this is important for the both for the research. I mean, if you want to make an infection, you want to know the concentration of virus in the sample. We call it virus titer, and that can be measured either by plaque assay, by number of plaques. But number of plaques is characteristic rather for plaque-forming units, and the number of viruses which infect every cells, we call it multiplicity of infection. And you already know what plaque is. I will show you more pictures of plaque. So multiplicity of infection, plaque-forming units, and also tissue culture and infected those. These are all helpful in determination the concentration of the virus, and it is important when you make an experiment, but it's also very important when you analyze the sample. For example, some sample taken from the body of the patient, and you want to know how much virus particles is present in the plasma or in the tissue sample. The technical terms which will be used very often are permissive cells. That means very often you already understand that the cells which create a microenvironment helpful for the virus to get in. So they have receptors, they have all kinds of transcription, translation factors specific for the species, which is specific for that pathogen. So, for example, bovine cells will be very permissive for bovine virus, but some viruses can, some cells can be permissive for many, for example, Vero cells, which we use very often. There are cells from monkey, and they are permissive for many kind of viruses, but many viruses also are very, very well, I don't remember the word specific about the cells which they can use for the propagation. And ELISA, I will talk about ELISA at the end of this lecture today. Some other technical terms. Monolayer, that means jednowarstwowa, one layer cell culture on the solid solid surface, and this is some. And I'm sorry to say that in virology lab nobody says I have to prepare jednowarstwowa hodowla komórek, but everybody would say, oh, I have to prepare 5 bottles with monolayers, because this is very, very obvious term used in virology. And this is, of course, monolayer means that cells grow on the in the vessel, plastic vessel, in one layer of cells, but they also can use sometimes different type of cell culture, mostly in suspension. We very often use the term FCS, fetal calf serum. This is a serum from cattle, and this is the most common addition to all media, which contains all the wonderful components which I mentioned during my lecture, of the growth factors, hormones, and so on. And the quality of that part of the medium is crucial, essential for the culture. So it also has to be very, very well protected. This is very rich component, and it has to be protected from the. So it is always kept frozen, and only it is thawed when we have to add it to the medium. Monolayer is characterized very often by confluence. So if I show you pictures of the cells which completely cover at the bottom of the culture vessel, so there is no space left, and that means that it's 100% confluence. If we have 100% confluence, usually this is not a good moment for infection with viruses, because there is no space for the cells to grow, to divide, and that means that the cells may be quite old, and that it's not what virus likes. The virus likes nice young cells. I'm afraid they also like young bodies. This is why children, for example, in the preschools, are so immediately infected when one of them is infected. But in the laboratory, we try to prepare cells with about 70% confluence. The word inoculum, you probably know. This is that small portion of the virus lysate, of the virus stock, which we use for infection. So usually we know how much virus and how much virus particle is that, and we have to incubate monolayer with in proper medium, in proper level of growing in the cell with very well specified amount of virus in a column. And just one word which we will be using very often is transfection. It's a bit like transformation, but transformation is either used for bacterial cells. Then by transformation, you understand, you know transformation. You probably did experiments like that you introduce plasmid DNA into bacterial cells. If you do that for animal cells, for example, we call it transfection. That means that DNA or RNA of the virus is introduced into the cells. Okay, so that's I will come back to those terms which are connected with amount of virus particles. So multiplicity of infection. Very, very crudely, you can say that this is a number of virus that are added per cell during infection. So if 1 million viruses are added to 1 million cells, that MOI is 1. If 10 million viruses are added, then MOI is 10. And if it is only 100,000 2 million, then MOI is 0.1. This is not precise, because, for example, if we add 1 million virus to 1 million cells, that doesn't mean that precisely in one viral particle goes to one cell. Maybe one cell which will be hit by 10 viral particles, and there are other cells which will not be infected with any particle. So this is, let's say, the number, but that doesn't mean that each variant, each infection will be ended with which will lead to formation of plaque. This is the formation of plaque. I don't know, it is on the next one. The formation of plaque, this is a number of virus particles capable of forming plaques per unit volume. So again, here, these two terms are very closely related, PFU, plaque forming unit, and MOI. It is has to do with a number of virus particles, numbers of cells. But here we just try to understand how many viruses we use in our experiments. We know that if we use very, very high MOI, I mean that there will be a lot of viruses per each cell, the probability that each cell will be infected is very high. If there is very low MOI, then there is very good chance that we'll see single plaques because only single cells will be infected. Okay, that can be also mathematically more precisely defined, but I don't think we need that kind of precise calculations. Okay, the other. So if our virus is causing cytopathic effect and we can observe the plaques, then we can use those methods to calculate how much viruses we have in the samples by plaque formity units and by MOI. But if the virus does not cause any plaques, I mean it is invisible in normal culture, and then we have to use more indirect method, and this is called tissue culture infection dose 50. This amount virus particles required to obtain infection in 50% of inoculated cultural cells, and this is called the CD50. It may be also infectious dose if it is 80-50 if it is used, for example, infection of animals. But I will come back to how to put that TCID50 in further parts of the lectures. So practically that will, this is in English, yes. So the day before infection, if we want to isolate characterized virus, we have to prepare a fresh monolayer, a fresh layer of cells of the permissive appropriate cells, and we can do it in a bottle or in the plastic well. And next day we examine the cells under the microscope. We check the confluence, how many, if the bottom of the vessel is covered to a larger extent by the cells, then we have to make sure there's no contamination of some kind of suspicious other pathogens present in our sample, and then we can infect. And oh my God, in Polish. Okay, I will try to talk very, very slowly. So after we we have the cells prepared, we remove the medium, so there is no dilution of our sample, and then we add that inoculum, that small sample containing viral particle, in such a way that that whole surface will be so we remove that medium, then we add inoculum, that means sample containing small sample of medium containing viruses, and we try to make it that volume very small, just to make sure that each virus has good close contact with cells. And then we leave that virus for a while, an hour or longer, and usually then sometimes we incubate it for 1 hour and 37 degrees, sometimes for a longer time, and then we move that bottle just to make sure that it is never dried, and then we remove that inoculum. Of course, whatever is left, it has to be put into sterilization, just the contamination. And then we add to that. So after removing that inoculum, we add fresh medium, and then we wait. Until we'll see, for example, something like that, the single plaques, which we obtained when the multiplicity of infection, the amount of viral particle were low, so not each cells were infected, or if that was very high MOI, then we can expect that all cells will be infected. For example, if we want to study the plaques, then we'll use that low MOI, a small amount of viruses, viral particles. If you want just to propagate the virus, then we use MOI quite high from the beginning of the experiment, and we collect then the cells. Is it shown further? Collect the cells just to obtain viral stock. Well, the vial is that solution when we have a lot of viruses, when we can freeze and keep it for further experiments. Well, how do we can we isolate and purify viral particles? So if we collect that medium from let's imagine that it has been already infected, then we have seen something like that. Then we collect that medium, and we also because some virus particles were trapped inside the cells. So we freeze and thaw infected cells to release those trapped particles, and then such fluid has to be centrifugated and remove all the leftovers from the cells, and then again second centrifugation to obtain the pellet. That pellet contains the virus, and then we can purify the virus, for example, by gradient centrifugation. I'm not sure if you ever have been already acquainted with, well, we've seen how and that works, but I'm sure you know that there are cesium chloride or sucrose gradients. Gradients can be used to separate the virus particle from leftovers from the cells, from the empty viral particles from the other contamination, and then such a band can be removed from after centrifugation and used for further investigation. Okay, that's about how to obtain viral viruses for our work. Well, how to characterize the virus. Well, perhaps that would be okay. I will go to that name. I'm not sure, but some of you perhaps worked with bacteriophage lambda, and then the most classical virology experiment is so-called one-step multiplication curve. What does it mean? You want to see what amount of virus is produced per cell and per the time, and that curve look like that. What is characteristic for each virus is how many hours after viral absorption, after start of infection, how many viral particles are released. And for some viruses, for most of viruses, there is something which is called eclipse period. That means from the incubation with the virus, for a while nothing happens. I mean, because the initial steps of the viral production are started, but there is no infectious particles. But gradually we can detect the appearance of infectious particles, and then for each viruses, the important information is how many viruses are intracellular and how many viruses are extracellular. And that is different from non-enveloped and enveloped virus. For non-enveloped virus, you can detect quite a lot of intracellular particles which are infectious, because if you freeze and thaw the cells, the virus which is released from those cells would be infectious. In the same time, of course, extracellular virus, the virus which is which goes to the medium, is also detected. But with enveloped virus, what is inside the cells, which has not been expelled to the outer space, that is very, very, very low, because intracellular virus, that means that there are viral particles which are not completed. They do not have usual envelope. They have to go through cell membrane to gain that full viral membrane. So this is viral envelope. So this is different. So all these factors, I mean, how many hours after absorption there is, if there is more intracellular or extracellular virus, how long is that eclipse period when there is still only so no virus is produced. These are ways to characterize the virus. So again, is what extracellular is that virus which is made and released from the cells, and total virus is extracellular, and total virus means the total number of produced viruses, intracellular and extracellular virus. This is how we measure that. Okay, these are all very technical, but just to now you have to what is important is that the virus yield, that means how many particles we obtain from during that cell cycle. It's different for different viruses. Poliovirus has replication cycle of only 6-8 hours. herpes simplex, about 24 hours. But for example, cytomegalovirus is another herpes virus. It's as long as at least 70 hours to obtain maximal growth of the virus. So when you work with certain viruses, you have to know how long you should wait with your cell culture to obtain as much viral particle as possible. Okay. About the virus detection. If you think about cultural cells, they're like two-dimensional animals and infection, and seeing the cytopathic effect, you have to find all ways to detect the virus. So one of them is, okay, going to beginning against from the diagnosis. Of course, I mentioned already all kind of clinical observation, but I'm now talking about detection and quantitation of that means evaluation of the number of viral particles in virology. So you can detect the viruses as physical particles, and for that is live infectious virus is not required. You can detect them as macromolecules, that means nucleic acid or proteins. Here a live virus is not required, and some biological agents that live virus is required in plaque or POC assay. And instead of detecting the virus, you can detect the antibodies against the virus, and that confirm the infection. So detection of virus of physical particles, this is already discussed, that there are all picture of the viruses done by electron microscopy, which is of course can be used to detect the virus it is present in certain kind of simple, but of course it is not a routine clinical virology laboratory. But for example, some viruses have very, very characteristic shape of the viral particle, and maybe helpful in detecting the virus, and maybe helpful to characterize it. But when it is used when we have sudden outburst of a certain disease. Yes, let's say that it is Africa and we have something like Ebola, then people will try to take the samples and definitely will try to look at the electron microscope just to see if they see any familiar shape. Of course, electron microscopy is also possible to use as an with characterization with the exploitation of immunological features of the virus. So it might be immunogold, and that was helpful. And of course, at the moment, or the modern world, it is only helpful, but the basic test will be the analysis of the sequence of the nucleic acid. But still, this is very, very important, because you can see that in many samples and some so the diagnostic, this is not routine diagnostic tool, but it's helpful in the diagnostic. So electron microscope that requires skilled personnel, that requires expensive equipment, and sensitivity is comparatively low. So if you have a sample when there are only few viruses, you may not notice that in the sample at all. And so for example, rabies, rotavirus from fecal samples. These are examples of when the electron microscopy used for the confirmation that is that kind of virus. And as I mentioned, that may be also immunoelectron microscopy. This is usually when the antibody was labeled with gold and solid. Microscopic techniques are used for the detection the virus with the gold labeled antibodies. But as I'm saying, this is not diagnostic tool. This is to characterize the virus. This is to see how the virus looks like. But generally, what we can detect in the clinics, parts from the laboratory, we can detect viral proteins, which are antigens. We can detect viral nucleic acid. We can detect infectious virus, and detection of the antibodies against the virus. So we maybe okay, we can say that some texts are direct or indirect. Direct method that we really detect the virus by isolating the virus, by genome detection, by antigen detection, or we can detect in the patient, in the serum of the infected organism, antibodies against the virus which are trying to identify. And this is really for many diseases, many pathogens, this is the number one method. Here, if you think now about the present pandemic, what is used as a specific test to detect the coronavirus SARS-CoV-2 presence is genome detection by PCR, real-time PCR, which is very, very specific, and we can not only detect the virus, but also how much of the virus, how much of the virus genetic material is present in the sample. And for example, this is also to quantify how much virus, how much nucleic acid is present in the sample is, for example, very important for HIV. HIV might be also quantified by the evaluation of RNA amount, viral RNA amount in the blood of the patients, and then according to the situation, the therapy, which is at the moment perfectly available, can be applied, or maybe another drug introduced into therapy, or more, just more drug. Virus isolation in case of coronavirus is a problem because it requires very high safety level, but in many cases it is very, very useful because, for example, you get a sample when there is only a few viral particles, and you have to use them to infect the cell culture, see the plaques and so on, but also obtain more material for further research. And antigen detection, again, coming to coronavirus, you know that apart from PCR, there are plenty of antigen tests, which are constructed in such a way as to detect the presence of virus proteins. Usually it is N protein for coronavirus, nuclear protein, which is the most abundant protein in that virus. So all these methods are in use, and then I will just try to concentrate first on the identification and detection of viral proteins. And if you see the names of that methods, they all start with immunofluorescence, immunohistochemistry, immunoblotting, Western blotting, immunoenzymatic assays like ELISA enzyme, like immunobioluminescence, and there is also immunochromatography, immunodiffusion. These are not very common methods. Immunofluorescence, let's stay with that 4. Okay, in Polish. Okay, let's go to the immunology detection directly. So immunohistochemistry, what does it mean? That means that if you have a sample, sorry, what time is it? I don't have any clock here. Should I make a break, or is it, who knows the time?
It's almost 11 o'clock.
Okay, in a minute. Okay, so let's go a little bit further. And so immunochemistry, immunohistochemistry means that we have a sample, for example, a piece of skin, a piece of biopsy from some tissue, and we can fix it with chemical reagents, and then incubate it with antibodies against the antigens of the virus. We suspect this is the virus, and then antibodies can be connected with an enzyme which is responsible for color reaction. The other ways would be to incubate it with antibodies which are directly linked to a fluorochrome. Then this is immunofluorescence, and then our sample will be shining in the fluorescent microscope. So this is example of herpes virus antigens in neuron intragenal ganglion. Here in endothelial cells. Of course, that may be made on the same principle as in Western blot, in which that means that we have 2 antibodies first, and the primary and secondary antibody. We come back to that. So this is another example of immunofluorescence from our laboratory. You can see that we can use, it's not only detection, but we can also confirm that, for example, we have working with a mutant which is which was some gene is deleted. For example, if we here gene which is called GI, that responsible for coding for glycoprotein membrane GI of fluorescence of herpes virus was not present, so there is no reaction with here on the opposite. Here you can see that there was no one of the glycoproteins, and the and the antibody against the glycoprotein 1 and glycoprotein work, against other that didn't work. So this is not only the matter of detection of virus, but the detection also the specific antigen, specific mutants. Another examples of immunofluorescence experiments. These are insect cells which produced viral antigens and connected with GFP, and that was very easy to detect. Here you can see viral proteins in infected cells, immunofluorescence, and so there are red. And here, for example, it's a example of liver cells where hepatitis C virus is replicating, and then you can see the complexes green in a liver cells. This is also stained with specific antibody. The other method which you, I'm sure, are familiar with to detect viral antigens is water blot on immunoblotting. So you analyze the sample which contains the virus so you put antigens on the gel, perform Western blotting, perform the electrophoresis, then Western blotting, and then analyze with the specific antibody what you can see. So you can detect the viral antigen, but also you can detect mutation. This is already research purpose of the experiment. This is also from our laboratory. So we have analyzed all kinds of mutants which have modified or somehow have deletions or some other changes introduced in one of the glycoprotein genes C, and you can see that the main band may be sometimes few bands, and may also, if we add GFP to that gene, that the band was bigger. So this is also for research purposes. Got another type of Western blotting. This is from another animal virus. Here we analyze the level of glycosylation, so you can see that the same protein from various viruses had a little bit different molecular weight depending on the how much glycosylated, how much glycans were added to the protein. And this is typical or the diagnostic test when, for example, and here the same. Sorry. So this is when you analyze the samples with known antigen, or you analyze you just detect if the serum from the patient detects the antigens of HHV, which you separate on by electrophoresis. And this is more about immunostaining of insect cells. You can see that, for example, here we have insect cells expressing a protein. It is from the parasite Fasciola hepatica, and here you analyze both the expression in insect cells. The red cells means that the cell was producing that protein, and we check the same by Western blotting. Okay, and so perhaps I will make a short break now, and I will meet again in, well, about 10 minutes, okay? Okay, thanks. Mm-hmm. Okay, I'm back. Are you back as well?
Yes, we are.
Okay.
Yes, yes.
So we are okay. We now finished with the viral antigen. So in the other way to detect and identify the viruses is to detect and identify its genetic material, nucleic acids. And here we have several methods available. The most popular is, of course, PCR, but the other ones, which also has its specific application, is hybridization, and you probably know the name of Southern blotting. I will come back in a minute. And of course, this is with the detection, identification, the other, apart from PCR, sequencing of the whole genome of its fragment is also very important diagnostic method, and the restriction analysis of nucleic acid. So let's start with that. Sorry, doesn't move. Why not? Perhaps we start with the Southern blotting or hybridization. Some of you will meet that method during your studies, but the principle is like that we have some viral DNA, we have to digest it with restriction enzyme, label with radioisotope, with phosphor, usually it's radioactive phosphor, and then we can incubate it with a probe, which I mean that fragment is a probe, and can be incubated with the samples we do analyze, and finally we make an electrophoregram and check if nucleic acid from infected cells or if not infected cells will be reacted with that probe. This is one. So the whole thing is based on obtaining a radioactive probe, and of course we have to know what kind of DNA we're expecting to prepare to use the probe with the known sequence, and then separated by electrophoresis fragments as analyzed by filter membrane. And what is the benefit of that kind of analysis is that, for example, here you can see that we analyzed a fragment from pseudorabies virus with many strains isolated from field isolates from animals, and you can see that there are we use not radioactive probe here, but bioattenuated fragment of nucleic acid of that pseudorabies genome, and you can see that there are plenty of mutation in that area, which also has the diagnostic value. And here you can see that the other method connected with the analysis of the whole nucleic acid. This is restriction analysis, and for example, this is the comparison of genomic DNA of human herpes viruses. There are all herpes viruses, herpes simplex, cytomegalovirus, varicella zoster, Epstein-Barr, and other viruses, and they have been all digested with the same enzyme BAM-H1, and the result then the result of that digestion was separated and by electrophoresis, and you can see that there are some bands which are very similar in all the viruses, but generally that pattern is characteristic for each virus. And also another restriction analysis here. Again, we've been looking for a mutation which were present in strains coming from different countries. So although this restriction analysis, hybridization, Southern blotting, analysis with specific probes are methods very much limited to laboratory use, not to clinical use. They are very often still used in our research. I'm not going to explain what PCR is, polymerase reaction amplification of RNA using known primers. You know that very well. You can hear also about that method every day when you are talking, and you probably have been tested by in the way when somebody has a PCR test. Although the PCR which is used for coronavirus is different time, it's real-time PCR. That means that not only you detect the fragment, and here you detect the fragment. If you see the probe, there's a fragment which appears after incubation with the specific primers and several cycles of polymerase reaction. And then you can see the bend on the gel. But in a real-time PCR, you can see how that signal from the specific fragments accumulates. And then you can see not only that the answer is yes or no for the analysis, but also how much of RNA of, for example, coronavirus is present and it depends how many cycles are needed to obtain the positive answer. The problem with that kind of analysis, this kind of diagnostic, is that with normal PCR and sometimes with real-time, it's a very, very, very sensitive method. That means it's very sensitive also to contamination. So, for example, if you work in the laboratory which is focused on detection of specific virus, you have to be sure that the samples will not be mixed in any way. And I'm not mean mixing from tube to tube, but for example, if you you have to be careful that if you work with material from some group of patients, you will not open the tube, Eppendorf tube, because it may when other tubes are open, because that may happen that the aerosol present during the opening will get to other samples, and that could be false positive reaction. Well, in comparison of the two methods, because I'm trying always to talk about coronavirus, this method, PCR, real-time PCR, is much more sensitive and much more precise. There may be some false positive reactions because of the sensitivity of the method, but in comparison to antigen test, which is now very common, well, this is much more accurate test still, because to have a positive answer in antigen test, you need really quite a lot of the antigen. Well, here only a few copies of RNA or DNA for other viruses is enough to detect the virus. So the method is very sensitive, very popular, because it is now done in routine ways in many laboratories. But for example, in certain situation, it has to be repeated. At the moment, real-time PCR is really so routinely used and so much standardized that optimized that it is really still the best way to detect coronavirus, at least. And now I'm coming to the other methods. That means it is connected with detection on infectious live virus. As I already mentioned, sometimes it is important to multiply the material which you have obtained from the first sample, and it can be done in the cell culture. And for example, you can then see the plaques or morphology of cells infected, and it is already some indication what kind of virus that is. It can be, of course, done not only in cell culture, but also in embryonated eggs or even in infected animals, but these are, well, not the routine ways. And as I mentioned, the benefit of such analysis is to obtain more values. You can use it in further analysis. So I already mentioned that live virus, you can observe the characteristic cytopathic effect. This is also a diagnostic factor. All that kind of inclusion bodies or formation of syncytia, and the shape and size of plaques are really is something which can tell us a lot about the virus. For example, this is from our own laboratory. You can see that we analyzed the plaques on different type of cells. MDBK is Mertin Darby bovine kidney cells. This is from bovine herpes virus. This is EBTR epithelial cells from bovine trachea, and here are from, as far as I remember, from umbilical cord, and we have to, this is how white type, that means white healthy virus, forms that kind of big plaques. If we remove one of the glycoprotein genes, the genes coding for glycoprotein, the plaques are getting smaller, and the effect is visible with to different level on different type of cells. And here that was our study a long time ago, that mutation of this one tyrosine in that gene gives almost the same effect as removal of the whole gene. So as you see, this is the morphology of plaques, because then we were certain that the virus spread from one cell to another is inhibited by the deletion or that mutation. So this is something which is used also in research very often. And that quantitation of plaques, I already mentioned that there are those precise type of quantitative that find, okay, it's all about number of viral particles. I told you already that's important. For example, the level of viremia. You know that this viremia means that there is a lot of virus in the blood cells and it is spreading around the body. So it is very important to sometimes to know in clinic how much viruses is, how much viral particles in the body, in the fluid sample, in the tissue sample from the patient, but also, of course, when we are doing an experiments, we have to know how much virus we have. So the plaque assay is one of the ways to calculate how much virus we have. And this is also that size in morphological plaques are also very efficient, useful markers. But sometimes we do not see any plaques. The virus is not causing that kind of effect, and then we have to use antibodies to detect and quantify the virus. So I've noticed for many students that this is somehow confusing when I show something like that, because for many students it is much more familiar with bacteriophages and bacteria growing on agar. But this is actually the same principle. Here you have a nutrient agent and they have bacteria growing on that, and you add bacteriophages and you can isolate single plaques. That means bacterial cells which were killed by the bacteriophage, by the fact which is also a virus, but virus for which the host is bacterial cell. Here you can see the same on animal cells, which were being grown also on a round plate like that, in monolayer, and you can see that plaques are usually, in this case, they are bigger, but you can also here and here you can do the same. You can calculate the number of plaques. You can also calculate the number of pox if you are using that way of propagating the virus. I mean, using that embryonated eggs. But this is rather, well, not routine way nowadays, but although in pox viruses and sometimes influenza it is still used. But still that plaque, which is a very, very important phenomenon, and many methods and techniques in biology are based on the formation of plaques, which is here it's said that the plaque is the result of single infectious virus particle, although it's not quite precisely, because sometimes many viruses are entering one cell, and then as a result the plaque is formed. But still it is assumed that one infectious virus particle might be responsible for one plaque. And well, so plaques can be counted, like here is shown that, for example, here it's uninfected cells like that. Then infected, if we use different dilutions of the virus, you can see the single plaques. Here is the stains, the cells were stained with crystal violet. And how to do that? I'm sure you did something like that when you were counting bacteria or had bacteriophages. This is all based on precise performing of sample dilution. So we have, for example, virus stock, and we diluted well, in 1 to 10, 1 to 100, 1 to and so on, and then we obtain. If you ever do that, I know this is very difficult to explain something like that when I show it on paper on a slide instead of showing it in practice, but to do that precisely, it is important to change the pipette of the Eppendorf tip every moment, because if you are taking as little amount as 0.1 mil, you have to be certain that there is not much more left on the surface of the pipette, so you can use the new pipette, mix, transfer, and so on. To avoid error, you have to change the pipettes, and then you obtain something like that. This is titration of vaccinia virus on monkey kidney cells. And you can see that if the cell, we are using completely undiluted virus, I mean only 10 minus 2, that means 100 times undiluted, that we do not see the cells anymore because they are all killed. This is transparent plate. Here, when the virus were diluted 100,000 times, you can see the single plex, and then the higher dilution you can see cells here, and you can see already a lot of plaques, and with higher dilution you can see single plaques. I can show you this is from our own laboratory. Also titration is the same. You see here when there is a lot of virus, precisely all cells were removed, killed, and detached, and we do not see anything in the monolayer. There are leftovers monolayers. Here there's plenty of plaques, and here are single plaques according to dilution. Here is already the virus samples were too diluted, and we do not see any plaques. Again, similar experiment with growing number of dilution. So if you have a number of plaques like here, you can calculate how many viral particles were infected that cell culture, and you have to make the calculation like that. That there was usually what is given on such a well is 0.1, not full mil. So if you, for example, that it was dilution 10 to minus 5, that means the titer of the virus would be 10 to 5. That means 100,000 of viral particles, but it was in the volume 0.1, so you have to multiply it by 10 because the virus titer is usually given per 1 mil of the samples, and that means that virus titer was 10 to 6. But if our virus does not got any plaques, the more undirected way to evaluate, to calculate the quantity of the virus is so-called TCI-50. I already mentioned that. What we are doing, do I have a description? I think it was here, but okay, it's not here right now. What you are doing, you prepare a lot of monolayers in small wells, for example, 90 well plates, and then you add the serial dilution of the viral stock, and you are coming to a point when half of them, 50% of these small wells will be infected. How do you know when they're infected? You have to incubate these small monolayers with antibody against the virus, and then stain with specific, stain with specific antibody or secondary antibody, like in Western blotting, and then you come to the border, like here. That here it is. It can be calculated that the titer of that virus is something like 10 to 4, because in 10 to minus 5 is already nothing infected. Here everything is infected, so that border value is taken into account. And of course, you still have to take into account what kind of volume did you use for the infection of that culture. And this is that 10 to 4 would be, let's say, tissue culture infectious dose. So this is a approximate only way of measuring titer of the virus, but it is surprisingly precise if you repeat it many times, because this is always done in several repetitions. So the other way to detect the virus is to present the antibody against the virus, and here it is several tests: ELISA, neutralization, immunoblotting again, and hemagglutination. And perhaps I will start with neutralization. I have it. Okay, neutralization. This is a very simple test. You are hearing about neutralizing antibody all the time when people are measuring the level of neutralizing antibody against coronavirus. And neutralizing antibodies are simply the antibodies which attach to viral particle and make it unable to react with the receptors on the surface of permissive cells. So the basic way to measure requires the presence of the animal culture, cell culture. So we can imagine that there is a virus, and we incubate it with the antibody which is acted against the virus. So the virus is now look like that. It's all these proteins which are necessary to interact with receptors on the cell are covered with antibodies, and there is no infected cells. So that means that we have actually neutralizing antibodies neutralize the virus, makes it unable to enter the cells. If the antibodies do not recognize the virus, that means that this virus is still able to infect the cells, and we have CD infection. So this is, of course, has to be repeated, and then the antibody has to be diluted, and so on. But this is a very basic test. But well, this really requires live virus. So neutralization measuring is a challenging test when we have to work with very, very dangerous pathogens. And well, sometimes you have to remember that okay, this is how to usually done. There is a serial dilution of the serum, incubation with the virus, and then you can measure which dilution of the sample prevent the plaque formation. So this is neutralized. Okay, I think I skipped that. So another method which is important for in virology is hemagglutination, and it also well that works in different ways. You can detect the virus, you can detect the antibody against the virus. What is the principle of that method? The principle of the method is like that normal, perhaps is shown here. Yes, if you have solution of red erythrocytes, red blood cells, if they stay in the test tube, after a while they will precipitate and make such a pellet. If you add a virus which has an antibody, which has an antigen with causing agglutination, claying of those blood cells together, agglutinate, then you will see something like that. There will be no pellet. There will be that kind of big complexes, which looks like that on the mold plate. So that is a method which allows us to confirm the presence of the virus. Or on the opposite way, if you want to be sure that the antibodies present in the serum of the patient contains antibodies against that virus, you combine first the virus with the antibody, and then means that that virus is unable to cause the hemagglutination, and then there is against inhibition of hemagglutination. So you can detect the virus, or you can detect the antibody against the virus. So just the description to make it more clear. It started with influenza. Perhaps you know that the main protein in the envelope of influenza virus is called hemagglutinins because of that specific ability to cause agglutination of red blood cells. And so this is a description how this has been done. For example, well, this is very classical and very, very still very much used in clinical laboratory, clinical laboratories method, but in research laboratories is perhaps not that common anymore. And it is how that looks in practice. So you have that multi-well plate, and you can see that when the virus was not diluted and incubated with the red blood cells, we can see that agglutination of blood cells. It looks like that. When there is very high dilution of the virus, you can see that pellet, such a button in the bottom of the well. So that perhaps again you can see the agglutination and no agglutination, and it might be modified, as I said already, to determine the level antibodies. And here you do not use the dilution of the virus, but dilution of the serum of the antibodies. If the antibodies contain enough the serum contains enough antibodies to neutralize the virus, then it can inhibit hemagglutination. And again, this is hemagglutination inhibition. Here you do not use this constant amount of virus, but dilution of the serum. Okay, I hope you understand the principle of hemagglutination, and this is against, this is another Western blotting showing that instead of detecting the viral proteins, you can detect the antibodies against the virus. So, for example, here antigen was the purified cytomegalovirus, and it was incubated with the serum from the patient. If the patient serum contains antibody against that antigen, then you have a positive band, which is shown here and here. This is cytomegalovirus glycoprotein one, and that brings me to ELISA. Which is the name of the enzyme-linked immunosorbent assay, enzymatic test, which has about hundreds of different flavors. Non-competitive, competitive assay, direct, indirect, double antibody, and I think it is one of the most common methods used in the clinic, although it's not as precise as, for example, real-time PCR. It's not that quantitative, but it's a method which can be used on big scale. The simplest principle of ELISA is like that we are covering, you are using a plate like that, multi-well plate, and you cover that plate with a non-antigen, which is attached to the surface of that microtiter well. That plastic has to be of special quality to make sure that the protein is attached very, very precisely to that well, that it is coating the whole well, and then you add the serum containing antibody to that antigen. So, for example, this is a virus like here. Then you add, for example, virus-specific antibody, maybe human, maybe rabbit in this case, and then you wash. If these antibodies are linked, then usually it may be like that, done like that this antibody is already connected to an enzyme or fluorochrome. Then you can directly see the results. But usually, what is being done is that double reaction, that first antibody is also an antigen, is an immunoglobulin for the secondary antibody. For example, here the secondary is the goat anti-rabbit antibody, and then it is usually connected, linked to phosphatase or peroxidase and enzyme, which is which later converts substrate, which is added to that well into color reaction, and then the intensity of color can be measured. And well, the problem is with that. So this is perhaps two versions of that test. So we have, as I showed before, this is virus, whole virus or virus antigen, antibody in the sample, and then anti-immunoglobulin secondary antibody with the indicator for ochrome or the enzyme. Usually this enzyme, and this is why enzyme linked immunosorbent assay. Sometimes if you have very little antigen which you want to analyze, you can use the capture antibody. So actually what is coating the well is not an antigen of the virus, but antibody against the virus. That is the way to detect the virus in the sample, and then you incubate it with the virus, and then you use another antibody, but that antibody has to be recognize different epitope than that first, which were used for the capturing of the antigen. So this is another approach to detecting the virus. Here you detect rather the antibody against the virus. Here you detect the virus. Oh, I will not be probably explaining all of that. There are many competitive methods. That means that before the incubation, antibodies are mixed with other antibodies just to enhance the reaction or to inhibit the reaction, and well, maybe direct method when the first prime antibody is already connected with the enzyme of from secondary antibody when they're using with the second antibody. Well, what is the problem is, for example, let's look at that little well and whatever is capturing antibody and the virus and so on. The whole method is based on the fact that the protein, in that case antibody or virus, is a protein which is sticking to the walls of that well. So the first step is to cover it completely. If it is not covered completely, then the next factor in the reaction, that means it is also, for example, an antibody, can fill in that holes, in that empty spaces. So the first thing is to coat it completely, then remove it completely, wash. That washing is always in ELISA crucial. Then incubate with the next step, whatever it is, antibody. Then again, washing, and then the next step. And very often in ELISA, you will not obtain black and white result. There will be something in the middle because there is a weak reaction. There are some leftovers which has been unwashed of the antibody. So ELISA is not such a wonderful method, but it is hard. So you can see that it will be colored somehow in the middle. So what we usually use is so-called cutoff values, below which or the values are treated as positive or negative. That it doesn't make sense to explain that right now, but we also very often use that methods in the lab, not only to detect the virus or to detect the antibodies, but to detect interaction of proteins. For example, if two proteins interact with each other, we can cover the ELISA plate with that protein which we want to analyze. Then we incubate it. Of course, wash the leftover, then to be antibody and antigen, although this is very strong interaction, then wash again, and then we try to detect if the second protein is detected by the specific antibody. So this is very often method used to detect which proteins form complexes together. So again, it's just showing. Well, I can skip that. Just once again to different. This is example of the enzyme horse radix peroxidase and substrate. Just to make it, this is called the sandwich ELISA. So there are different levels: capture antibody, target protein, detection antibody, and another secondary antibody. So as you see, there are many levels when you can, if something is done not very precisely, I mean, if the capturing antibody was not capturing enough protein, or there was some uncoated uncoated space in the well, and so on. So there are many ways to have false results here. Okay, I already mentioned that. I mentioned that as well. So just to summarize, we can always detect by enzyme linked, and so also immunosome or the assay by ELISA, either viral antigens in patient sample or antibodies in patient samples. And the principle is similar, just what is first used to code the well, it's different, but this is usually ended with the measuring the intensity of the color in all those wells. It has to be always done in several repetitions to make sure that we didn't make the mistake in the pipetting and so on. So if you now compare the techniques which I mentioned today, what is the limitation, what is the cost, what is the principle, and so on. So viral plaque assay is relatively inexpensive, but requires a lot of work. Sometimes it takes days to obtain the plaques. Here, when you don't have to wait that one with TCD50. So even if the plaque is not formed, but virus already infected the cells, this is can be done. Well, the mentioned PCR excellent, not very quick, moderate labeled. Well, it is supposed to be expensive. It's not anymore that expensive, although real-time PCR, the probes which are used are usually expensive. And you know how it works with immunoblotting. Reproducibility is relatively good. The same with ELISA, but I didn't make about cytometry, but transmission electron microscopy, they are not really diagnostic methods. Okay, this was, I think I will stop that because I just mentioned that I was talking all the time about because I was trying to talk about methods to characterize the virus, identify, detect, identify the virus. But how we work in the virology lab, it's a bit different because we very often have to obtain viral mutants, and that's quite difficult procedure. And perhaps well, only if you start working in really virology, you know that why do we want to obtain viral mutants? First of all, if you want to, for example, understand the function of a specific gene in the viral genome, to somehow to delete or modify that gene would be seeing for the phenotype what is the effect of the virus life cycle on viral growth and so on. This is one of the basic methods. On the other hand, we need mutants sometimes as vaccine strains, for example, and this is or vector vaccines or vector strains. So that's really a huge part of virology, how to obtain, analyze a viral mutant. And classically, it was mutants were done by spontaneous or induced mutant production, and that was such classical methods. Well, I will be talking that on the lecture about vaccines that involved, for example, passages by a lot of cells, sometimes unusual for that virus, different temperatures, exposed to UV and other mutagenesis factors. But of course, it's uncontrolled, and in the modern laboratory, we do not use that kind of methods. We use method construction by genetic engineering, and this is relatively, no, it's not very simple, but with DNA viruses, it is possible. With RNA viruses like corona or influenza, first we need to obtain a cDNA copy of the virus. If we have cDNA copy, we can manipulate the DNA easily. With RNA, it's not that simple. The problem with obtaining mutation in the viral genome is some genomes, like for example herpes viruses we work with, are very large, not like plasmid, 4 kilobases. We have 140 or 200 kilobases, and then we want to introduce one mutation, so we have to do that in different ways. And definitely what we use then is, I already mentioned that term which is called transfection, and transfection means that we introduce DNA into a cell. To obtain a mutant, what we do is we introduce to a cell genomic DNA and a plasmid. I don't know if I have it here. Like here, for example, we have viral DNA and we have plasmid with the fragment of the DNA, which is flanked by the other sequences. So we have fragment when we introduce some mutation. For example, we added GFP to a gene. It is flanked by sequences from that genomic sequence. So what is happening if we doing co-transfection? So we add DNA and that plasmid together into a cells, there will be a combination occurring in some not large percentage of viral particles, of the genomic sequence, and then we can, in very, very time-consuming and work-consuming work, isolate positive cells which carry that mutation. And then finally, we have to purify such a mutant from single cells and then confirm the presence of the mutation. We also used in the present the cosmids, that means large, for example, herpes virus, pseudorabies virus could be obtained as a collection of big plasmids, which are called cosmids from cohesive ends of lambda phage. This is as you can see, this is from 1988. This is a method which has been then elaborated, but it has been used for a long time. So you can introduce a mutation into one of the fragments, then you could put them together in a mixture and ligate them, and then the whole genome now contains the mutation which you wanted to obtain. Typical methods, this is called use for adenovirus. This is the method how to obtain adenovector. The adenoviruses which are used to production, for example, of AstraZeneca vaccines against coronavirus. This is done like that. That a lot of things are done in bacterial cells. A plasmid is prepared with fragment of adenovirus genome, and then recombination in plasmid is done with the whole genome inside the bacterial cells, and then it's that from bacteria it is that genome containing already the mutation is transferred to complementing cells, passages of the virus, large scale purification. Well, I know this is not clear for you. I'm just showing. I wanted to show you is that there are many different methods to obtain mutants, either because we want to understand its function, function individual genes, or we want to obtain, for example, vaccine vector. And these methods are very advanced nowadays, and also very advanced are the methods of final step, like, for example, virus characterization, stability for adenovector, stability, productivity, sequencing, transgene expression. This final product is very, very well controlled. And the same system is in baculovirus. In baculoviruses, we use for production of proteins in insect cells. Against the main recombination, like in adenovirus, is made inside the bacterial cell. And but this is something which is a little bit too complicated perhaps for today. So let's stop right here, because I mostly wanted to make you familiar with methods which are used both in the laboratory or in the clinic for detection and identification of the virus, pathogenic viruses or viruses which you want to work with. But of course, there's the whole large spectrum of biological methods which are used in addition to those basic diagnostic methods which can be also applied in the clinic. So I know this is very difficult because I'm talking about practical matters. I'm trying to show you a lot of pictures, but it is not the same as touching the pipettes, the bottles, the monolayers with your own hand, although please don't touch monolayers with your own hands. I mean, well, performing this experiment by yourselves. Some of you will be doing that during virology classes. For other people, if I had more time, I would show you more videos. But if somebody is interested, you can find a lot of video how to make transfection, how to isolate viral DNA, and so on. So that's all for today. And from next lecture, I will start to talk about individual virus families. And the first lecture is always begin with the beginning, that I will be talking about picorna viruses, representative of each is poliovirus, but also rhinovirus. And foot-and-mouth disease virus, and I started with poliovirus because that virus is for animal virology, for eukaryotic viruses, as important like lambda-phage for bacteriophage biology. It was a model for many years, and it was one of the first viruses which people investigated. So next lecture will be about picorna viruses, but I look at the calendar now and I see that next Thursday is is 11. Yes, that means it's a holiday. Yes, am I right?
Yes. Yes.
Okay, so this is a problem. Okay, let's go, because that means we are losing another lectures, and there is no chance that I will be able to I will have to select which lectures can be introduced, can be given during that program. But perhaps we just skip one or two, or we try to squeeze two virus families into one lecture. Anyhow, so that means that we are meeting not on 11 but 18 November, and I will be talking about coronaviruses. And until now, then that's all for today. Any questions? Okay, no questions. So thank you for your attention, and I will see you in 2 weeks. Bye bye.
Thank you
Thank
very
you.
much.
Bye.
Bye bye.
Thank you.
Bye bye.
Thank you.
Thank you. Goodbye.